All right, today we're going to pull back the curtain on the genetic secret that powers the entire multi-billion-dollar hybrid seed industry. It's an incredible story about a tiny genetic conflict happening inside a plant cell and the totally ingenious way that breeders figured out how to control it. So let's just dive right in. For any plant breeder, the ultimate goal is to create a hybrid that is way better than either of its parents. But how do you actually do that? How do you make absolutely sure that your high-yielding plant gets pollinated by that other disease-resistant parent without it just pollinating itself by accident? Well, the answer is hiding in a really fascinating natural phenomenon. And that phenomenon is called heterosis, or you might have heard it called hybrid vigor. This is basically the magic behind all of modern agriculture. You cross two different parent lines, and you get this offspring that is just dramatically more productive, more robust. But to make that happen on a massive scale, you need a totally reliable way to control the pollination. And for the longest time, that control was the biggest problem. It was the bottleneck. Trying to stop a plant from pollinating itself was just incredibly difficult and super expensive. We're talking about a process that required just massive amounts of painstaking manual labor. So this brings us right to the core problem that breeders were absolutely desperate to solve. Let's call it the hybrid challenge. The traditional solution was, well, it was basically brute force. It involved going in and physically removing the pollen-producing parts of the plant. It's a process called emasculation, or if you're talking about corn, detasseling. I want you to just picture crews of workers walking row by row, plant by plant, carefully plucking off the male flowers. It was so slow, which of course drove up the cost of seeds, and you always had this risk of actually damaging the plant you were trying to breed. There just had to be a better way. And thankfully, there was. But the solution wasn't some new machine or a fancy new tool. It was a hidden genetic off-switch that nature had already invented. This solution is called Cytoplasmic Male Sterility, or CMS for short, and it's a naturally occurring trait where a plant just can't produce working pollen. It becomes the perfect female parent. It's biologically incapable of pollinating itself, but its female parts are totally healthy, totally ready to receive pollen from another plant. It's like nature's own built-in emasculation tool. And you can imagine, the discovery of CMS was a complete and total game changer. As the research puts it, it became a significant boon in the realm of plant breeding. It was the key, the absolute key to making large-scale, cost-effective hybrid seed production finally possible. So, what's really going on inside the plant to make this happen? Well, the answer is this incredible little conflict between two totally separate sets of DNA, both living inside the same plant cell. The easiest way to think about it is like a genetic disagreement. See, most of a plant's DNA is where you'd expect it, in the nucleus, but it also has this other tiny little genome tucked away in its mitochondria, the cell's power plants. And normally, these two genomes get along just fine. But with CMS, they're at war. Now, this slide here really helps visualize that conflict. On the left, you've got a normal plant cell. Everything's compatible, everyone's happy, you get fertile pollen. But look at the middle. Here, a rogue gene in the mitochondria, that's the CMS-causing gene. It messes with energy production, and it stops pollen from ever developing. The plant becomes male sterile. But the really crucial part is on the right. There's a special gene in the nucleus called the Restorer of Fertility, or RF gene, and it acts like an antidote. It can just cancel out that mitochondrial signal and bring fertility right back. Okay, so just to recap the genetics here, this whole thing kicks off because these weird new genes, they're called ORFs, they just pop up in the mitochondrial DNA. And because mitochondria only get passed down from the mother, CMS is a maternally inherited trait. These rogue genes, they specifically sabotage the super energy-hungry process of making pollen. But the nucleus can fight back with those powerful Restore of Fertility, or RF genes. So, armed with all this knowledge, plant breeders developed a truly ingenious method to control this fertility on-off switch. It's known as the three-line system. This system is the absolute backbone of the modern hybrid industry. So first up, you have the A line. This is your male sterile female parent. Think of it as the lock. It can't self-pollinate. Second, you have the B line, or the copier. It's basically identical to the A line, but it's got normal mitochondria, so it's fertile, and you cross the B line with the A line for one reason, to make more A line seeds. Finally, you've got the R line, the master key. This is your chosen male parent, and it carries that all-important RF Restore gene. So out in the production field, you plant rows of your sterile A line right next to rows of your R line. The R line does all the work, pollinating the A line. The seed that results from that cross is your F1 hybrid. Now, it got the CMS trait from its mother, but it also got the FR gene from its father, so it's going to be fully fertile and produce all the grain or fruit that the farmer wants. No detasseling, no manual labor needed. It's just brilliant.
Now, as powerful as this system is, it isn't perfect. But today, brand new technologies are giving breeders a level of control over this genetic dance that they could have only dreamed of before.
One of the big challenges is instability. Sometimes things like high temperatures can cause a sterile line to just suddenly start making pollen again, which contaminates the whole hybrid seed production. Another big issue is that breeders have really tended to rely on just a few sources of CMS. And that creates this dangerous lack of genetic diversity, which could make our crops really vulnerable to a new disease or pest. Traditionally, finding and breeding these restorer genes into a new line was a long, slow process. It took years, sometimes decades, of trial and error. But today, things are changing fast. Modern gene editing tools like CRISPR and this really specialized tool called mitotalons let scientists just bypass that whole long process and go in and directly edit the genes inside the mitochondria itself. And this isn't just theory, okay? Let's look at a fantastic real-world example from the research. Scientists pinpointed this one specific gene, called ORF137, as the culprit that was causing male sterility in a particular line of tomatoes. So what they do? They designed one of these mitotalons to act like a tiny pair of genetic scissors. It went right into the mitochondria, found that ORF137 gene, and just snipped it. And this perfectly shows the power of this technology because the result was a complete, total restoration of fertility in those tomato plants. They basically created their own restorer line with surgical precision. So let's zoom out. Why does any of this matter? Well, cytoplasmic male sterility is not just some biological curiosity. It's the engine that makes the entire hybrid seed industry go. It slashes costs for farmers, it boosts crop yields, and ultimately, it makes our global food supply more secure. It's truly one of the most important tools we have in agriculture, period. And that leaves us with one final, pretty critical question. We now have the ability not just to use nature's genetic off switches, but to actually design our own. As the pressures from climate change on our food systems just keep growing, the race is on. Can we master these tools to create the resilient, super high-yielding crops we're going to need for the future? And can we do it fast enough? Thanks so much for joining me for this explainer.